You got your gym shoes on today? Because we're going to roll. What I hope you saw was a lot of this is review. Do I have extra of those study guides? Thank you. Thank you, thank you. I hope you saw that much of this is review. There are some details that maybe you don't have. But this is where we left off Friday. The basic components of a cell. Plasma membrane, cell membrane, the cytoplasm with all the pieces there, and the extracellular fluid. They have this picture of a representative cell. Are you comfortable? No cell actually looks like this, just has all the pieces all on one cell. Okay? They're just showing you all the kind of things a cell could possibly have. They actually did better in this book than the other book. The other book had this weird, funky, mutant kind of cell that had everything you could possibly put in it. They at least chose wisely some of these pieces. So, let's dive in to the plasma membrane. Okay? A plasma membrane is a phospholipid bilayer, 2 layers of phospholipids with the tails pointed inward, the heads pointed outward, right? And what do? They define the cell boundaries. They govern interactions between other cells, like, hey, stay out of my space. This is my space, right? And they also control the passage of materials in and out of the cell. They allow some things to enter and some not to enter, right? Lots of pictures of the plasma membrane, and I like this one. It's very zoomed in. This is 100 nanometers. Again, that's a billionth of a meter. They've zoomed in, and you can see there's actually 2 layers, right? An outer layer and an inner layer, those phospholipid bilayers. Okay? And again, all of these slides are up on Schoology, so I'm going to skip a number of pieces today, just so you know. They are there for you. You can check out the closer pictures of them. The plasma membrane picture. This picture is here on page 81. You should be comfortable with this. And you realize when I say you should be comfortable with this, you might see this again, right? Good. So they're showing you what this cell membrane looks like, right? And they're showing you the different pieces that are there. So let's talk about some of these different pieces. One of the first things we'll talk about is membrane lipids. And most of the lipids that are in the membrane most of the molecules that are in the membrane are lipids, 98% of them, right? And of that 98%, 75% of the membrane lipids are phospholipids, which makes sense because that's what the membrane is made of. Are we cool? And of course, we talked about hydrophilic and hydrophobic, the heads being hydrophilic and the tails being hydrophobic. So they're moving water out and water in, keeping it out, keeping it in. They're zooming in. That's what these green things are showing you, the specific pieces zoomed in on it. The second type of membrane lipid would be cholesterol. Cholesterol, this helps make the membrane stiffer and more complex because those phospholipids, they can just kind of move all over the place, like shift and change shape. That's why a cell is not some hard or rigid something. It's changeable, it's movable. The cholesterol gives it more stiffness. The third kind of lipid in the membrane is the glycolipid. Remember, this is a combination of what 2 things? What does glyco mean? Sugar, carbohydrate, right? So it's a carbohydrate lipid, this combination of the 2 of them together, right? And that basically provides some anchor sites where you can put things on there. Excuse me. We've got membrane proteins. We just talked about lipids. Sir, question? Mm-hmm.
So when somebody says that they have high cholesterol, does this mean that something's wrong?
So no, they're talking more about the amount of fats that are in the blood vessels is really what you're measuring the amount of this certain fat in the blood itself. And by having too much cholesterol, it can build up. It can actually stick to the walls, which then narrows the walls, which makes it harder for the blood to go through. We'll talk much more about that in the cardiovascular system. But it's the same piece that these cholesterols are just embedded in the cell. So high cholesterol wouldn't be measuring that. It's measuring the amount in the blood itself. It's a good question. Good question. The membrane proteins, okay, we've kind of got 2 different pieces of the membrane proteins. One that are integral or transmembrane. In other words, they go all the way through the membrane. And their main thing that they're going to do is let things come in and out. These are the channels that allow molecules to move in. These are the receptors for receptor-mediated transport that move stuff into or out of the cell. The other type is the peripheral proteins, and these guys are just stuck to the outside. They don't actually penetrate through to the inside of the cell. They're just on the outside. And what do these membrane proteins do? Some of the pieces that we've mentioned there. Here's a picture so you can see this is the protein, the purple piece here, right? That are grabbing these carbohydrates and bringing them in. Or they're letting water come in. They're going all the way through. So some functions are that they are receptors, again, transport. They are cell identity markers. Remember I talked about briefly on Friday that you have, there's actually like a process that the cells look at to see, hey, do I know you? And they'll like sniff each other, in quotes, sniff each other like two dogs, sniffing each other. They're looking at these membrane proteins to say, hey, is that an Amar cell or is that an Alex cell? Oh, it's an Alex? Oh, let's kill it. It's an Amar? No, that's good. That's good. Are you with me? We good? So they're identifying the cells on the outside. And the other piece is they're adhering to other cells. So they can actually be almost like Velcro that will allow two cells to kind of stick together, again, to form membranes. You're with me, friends? Yes?
So you said that the internal proteins and the transmembrane
Go all the way through. Yeah, they're basically the same word for the same kind of protein. Yes?
peripheral proteins can stick to either side of the membrane, right?
Correct. They could be outside or inside. More likely they're going to be on the outside because they're interacting with things outside the cell, but there are some that are stuck on the inside as well. You're correct. You're correct. All right, here's a lovely picture that's there in your textbook on page 83 that talk about these different functions that are present there, breaking them down in detail. Jess? There are cells in our body that their job is to kill other cells, invaders. Sure, right, we'll talk much more detail about those. But that's part of how they see, are you a friend or are you a foe? They look at these membrane proteins. Good question. All right, let's keep rolling. You have these things called second messengers. This is another function or another type of these proteins that are there on the outside, or actually the transmembrane proteins. And what they are doing is they're basically acting as a relay between the outside and the inside. So here comes this cell on the outside. They call it epinephrine. This is in your textbook there on page 84. And this epinephrine is trying to send a signal to make the cell do something. But it can't actually enter the cell, right? It's not cool enough to get past the bouncer. You good? So it binds to this protein, this second messenger, and by binding there, it causes the release of this G protein that will come and activate another enzyme that will send a signal on its own. So the actual messenger cell, this epinephrine is a messenger to the cell telling it to do something, but it can't get through. You need the second messenger proteins to get the signal through. Does that make sense? It's like, when you tell your brother or sister, hey, go tell mom and dad that they are the second, they are taking this message. You should have delivered it, but you've delivered it to them, and they're moving it on. Okay? You have this thing called the glycolax. I did not include this on your study guide, but this is kind of these external proteins and carbohydrates that are on the outside of the cell, and again, they help to identify it. They have a number of different functions that are listed here about protection and about defense. Make sure you check them out. Again, not on your study guide, but worth being aware of. A couple other pieces of the surface, the cell surface, this plasma membrane. You've got extensions. You've got pieces that are extra than just a regular outside membrane. So the first one is called microvilli. Check this out. Are you comfortable I could draw two lines. Which one is bigger? Bottom, of course, duh. I could take this bottom one though, and if I were to do like this, the same length of a line would now be shorter than that. Does that make sense? So I have the same amount of surface area, but I've just squished it into a much smaller package, right? That's all microvilli are. Okay? So the cells in your intestines that need to absorb stuff, right? They need lots of surface area because basically things go through the surface into the capillaries behind it to be absorbed. Again, all the proteins and the carbohydrates and the fats you eat, they have to be absorbed into your body. So these cells inside your digestive tract have tons of these microvilli that give it more surface area. So even though my intestines are like 30 feet long, the actual surface area is about 10 times that much because it's got all these little microvilli. Question? Okay. Yeah, it increases surface area. That's the key piece behind microvilli. Question?
So are they like extensions or are they the actual surface area?
So this is the actual cells, but instead of them just being nice and flat, round, so if here's a cell, instead of being like that, it would actually be like this.
Okay.
Does that make sense? Same size of a cell, but I've got more surface area, more places where things can be absorbed. Sir? It is still the cell membrane. This is still the cell membrane. It's just kind of a projection going upward. Okay, next we move on to, oops, come on now, don't move on me, just slide. Sometimes this thing works well, sometimes it doesn't. Here's a picture of those microvilli, again on your composite cell. Another picture here, where you can see, here are the microvilli, right? The outside of the cell that have these little projections going up and down. Our next one we're going to talk about is cilia. Not cilia, cilia. Cilia, you recognize that, right? So cilia, again, are longer, they're hair-like, so longer than microvilli. And these guys usually provide some kind of mobility to surfaces. They are not the little flagellum that are on a sperm cell. These are in between the microvilli and a flagellum. So cilia, the main piece we think of cilia is in the respiratory tract. Are you comfortable that you've got mucus that line all the inside of your mouth and your nose all the way down into your lungs? All that surface area is coated with mucus. Why? Because we breathe in stuff. And we want that bad stuff to get caught in the mucus and get flushed back out. How does it flush it out? By the cilia. The cilia acts like waves. It literally just moves back and forth and tries to push that mucus back up and out. Think of when you're sick. You're always bringing up nice, tasty yellow stuff, right? Yummy, right? Okay? And you spit that stuff out. That's your body saying, hey, this is bad. Get it out of me. Are we cool? And the cilia is what does that process, what moves that stuff up. It's in other places. It's in the uterine tubes. It helps push the egg from the ovaries down into the uterus and then out of the body. It's in the brain. It's in the testes. There's lots of places these cilia are present. But again, the big one we think of is the respiratory tract. I keep going. I don't want to grab you. I just want to move. This is going to drive me crazy today. Okay, and here's that cilia action, right? So it's pushing, it's moving, and you see this mucus that's laying on top. These are good epithelial cells, simple columnar epithelial that's pushing this mucus back up your respiratory tract. Cystic fibrosis, I'll mention that. It was in one of the little reading pieces you had. This is a disease where that mucus gets really thick. And it's very difficult for your body to get rid of it. So they have to go through all of these exercises. They do some pounding, like literally beating the person on the back and on the chest to help break up this mucus and move it out. Is if they don't, it gets infected and the person can die from cystic fibrosis. I've known a couple of different people who have had this. Life expectancy very low. Again, because breathing is fairly important to live. All right? And if you can't breathe, if this mucus is getting stuck and preventing you from getting oxygen in and out, it makes it a very, very difficult time. All right, let's talk about flagella. This is, again, the third of our extensions of the cell membrane. And the flagellum, you understand. Again, think of a sperm, right? The little head, but then the tail is what we're talking about, this flagellum. And its main purpose is for what? Movement. It's all about mobility. It's the propeller that propels that little sperm along. And we'll talk more about this when we get to that stage. But in Michael Behe's book, Michael Behe is a biochemist who looks at the actual molecules that make up these flagellum. There is a straight-up motor inside these things. Like if you took the parts of a motor, like an outboard motor on a boat, the flagellum is like exactly like it. It has the same kind of setup to it. And his thought is that this is what he calls irreducibly complex. If I take out one part of this, the whole system breaks down. And I couldn't just gradually, over millions of years, put all these pieces together to make this. You'd have to have it all come together all at one time. This is one of the examples he gives of why it sure looks like cells were designed. They didn't just evolve.
So just like car motors have problems, does that happen to men?
Oh, sure. Sure. Yeah, you just get rid of that sperm, obviously.
Yeah, but I mean, like, is it possible for men to have health problems?
Oh, sure. Yeah, no, definitely. There can be, that's one of the primary sources of men not being able to conceive. I know men don't carry babies, but they certainly produce half of the pieces that make babies, right? So it's not always the woman who has problems conceiving. It can be from the guy as well. Yeah, definitely. No, only specific cells that have any of these three, microvilli or flagellum. Biggest one, respiratory system, respiratory tract. Yep. Okay. Any questions there? All right, let's talk about membrane transport. Now, I'll let you stand up for a minute. Membrane transport, and you're going to recognize these things. You can stand up if you want. That's fine. Get yourself a little bit of a break. Membrane transport, you've heard of these things, right? Where we're literally bringing things through membranes. And there's lots of different ways that we do this, right? Because remember, that plasma membrane is selectively permeable, right? Some things can come in, some things can't. And so therefore, we may have to let some things in and help some things come in or go out. We've got two mechanisms, passive or active. Passive, no ATP. Active, yes ATP, right? Simply means passive will happen on its own. Active means something's got to happen to give it the energy to make it work. So let's talk about filtration. That's the easiest one. This is simply where particles are moving through a membrane simply by pressure. So think of this as just a regular filter. This is what you all are, when you think of something filtering something, I think about when we're camping and you've got a filter and you're putting water through one end and you want clean stuff on the bottom end, right? That's what filtration is, right? So again, this is a pressure, physical pressure that forces fluid through that selectively permeable membrane. Some things go through, some things stay out. We cool? Brings water and nutrients into tissues and allows removal of waste from the capillaries in the kidney. Here's a good view of what that looks like. You realize, of course, maybe you don't realize this, that your blood vessels are not solid. Like the capillaries are not solid. If they were solid, nothing could get out or get in, right? So there's these little openings that allow some of these membranes to come through, some of these solutes to go out, some solutes to come in. It just depends on the pressures that are involved. We'll talk much more about this as we go. Simple diffusion is our next one. Simple diffusion is, oops, I went too far. Is the net movement of particles from high concentration to low concentration. I have a great way to remember this one, and it's terrible, but you'll laugh, but stay with me. Somebody passes gas, right? And it's highly concentrated right around them, right? What happens to that gas? It flows outward, right? It moves from a high concentration to a low concentration. And suddenly the person in the back of the room is like, oh man, where did that come from? Are you with me? That is simple diffusion. From a high concentration, it flows to a low concentration. And again, this is solute, movement of solute. Osmosis is the movement of water through a selectively permeable membrane. And please, that's a key piece. Osmosis does not deal with solutes moving, it deals with water moving. Most of the other stuff we're dealing with is some sort of solute, something dissolved in water or in our fluids. This is actually water itself moving. And how does it move? Water moves from the side where it is more concentrated to the side where it is less concentrated. Or if you want to think of it another way, because I gave it on your study guide in both different ways, is also the movement of water from less solute to more, or from more watery to less watery. Are you with me? So again, it's the movement of water from the side where water is more concentrated to the side where it is less concentrated. So here you can see water, solutes, right? So because there's more solute over here, more water is going to come because it's trying to even out the concentration here. We're trying to make both sides have the same concentration. Remember, concentration is a measure of solute for the amount of water present. So if I've got more solute on the side B here, let's add more water. That will dilute it and make it less concentrated. So we can have equal on both sides. There are specific pieces in the membrane. Nope, I missed them. These guys here called aquaporins. Let's look at the word. What does aqua mean? Water. What's a pore? Yeah, like a hole, an opening, right? Like in your skin. So this is a hole in the cell that allows water to go through. Again, anatomists are not creative. We just name it what it's called, right? So an aquaporin is this channel that allows water to move in and out, even easier than through the membrane via osmosis. Aquaporins. Okay, we continue on. There's a whole bunch about osmolarity and tonicity. I'm not going to give you any questions about that. This is a normal red blood cell in an isotonic solution. In other words, the amount of solution that should be, the solute that it should be, it's the same as the red blood cell. The hypotonic means I've got too much water. The hypertonic means I've got too little. And the hyper and hypo here in this case are talking about the amount of solute present. That's why they're saying that. So you see the red blood cell swells up or shrivels up, depending on it. Okay? And this is part of the problem when we are dehydrated, right? It actually affects our red blood cells. They can't function. They can't deliver oxygen as well when we're dehydrated. We're moving on to carrier-mediated transport. What is a carrier? Something that carries, good, yes, right? Something that moves things, right? What is mediated? Like a helper, right? Someone who goes between two people, right? And solves a problem. So in this case, I've got something that wants to come in to the membrane, into the cell, but it can't do it on its own. It has to have a buddy to bring it in, right? It's like you're hanging out with that famous person, and you can't actually get into that club, but if you're hanging out with them, oh, well, they'll bring you in, right? Carrier-mediated. So this carrier protein is helping along. And we've got a couple of pieces that go along with that. 3 kinds of carriers. No, that's not what I want. The 3 mechanisms: facilitated diffusion, primary, and secondary. Let's talk about them. So, our facilitated diffusion is where we are transporting a solute down the gradient. So we're going from high to low, right? Just like we did in diffusion. Are you with me? But it's facilitated. Again, it won't move on its own. It needs something to help it along. We cool? Again, the solute attaches onto a carrier, and the carrier moves it to the other side of the membrane and then releases it. That's our facilitated diffusion. Here's a picture showing you the same thing. Our primary active transport is where a carrier moves the solute through a membrane up its concentration gradient. So if you want to use the example of somebody passing gas, it would be like that gas, instead of spreading outward like it went back inward. I don't know, it's kind of weird. Okay? But that's the thought, is that these carriers like grab onto the bad particles and shove them back the other direction against the gradient. Does that make sense? That's our carrier-mediated transport, the primary active transport. And again, an example of this is our calcium pump or the sodium-potassium pump. We're going to spend a moment talking about that because that sodium-potassium pump is a great example of this. Each time this pump, it consumes 1 ATP and it takes 3 sodiums in and 2 potassiums out. It's a matter of keeping the potassium concentration higher and the sodium concentration lower within the cell than it is in the extracellular fluid. And the point behind this is it allows things to happen. It allows nerves to fire. It allows muscles to contract. I did not know this until I read this. Half of your daily calories are utilized just simply on this pump, on keeping these concentrations the way the body needs them to be for you to do what you need to do. I thought that was a very interesting fact. I'm not going to test you over that, but that was an interesting fact. This is a very important piece because calcium and sodium are always moving out, not calcium, potassium and sodium are always moving out and in, and we're trying to make sure our concentration stays stable. Are you with me? And again, that's primary active transport. The next piece is secondary active transport. And what this is instead of something purposely taking Sean and bringing him, so here's our carrier, our primary active transport. As I say, you know what, Sean, I really like you, come on, let's go into this club, you and I together, right? And I carry him in. But Zach's always hanging around Sean, right? And I say, yeah, all right, Zach, come on, you can come with us. So I'm not primarily taking Zach in, but because, you know, we're friends still, he can still attach to me and he can just come along for the ride. Does that make sense? Nothing against my friend Zach, of course. But that's the point behind secondary active transport. It's not actually meant to move that molecule, it just comes along for the ride. Are we cool? And again, this is active. You need ATP in order for these to do that. Because again, we're going up gradient. We're not going with the gradient from high to low. We're going from low to high. Yes, ma'am? That's a good question. I think it's active.
But it's transport down gradient, right?
I'm pretty sure facilitated is active. But let's make sure. Oh, it does not. So no, that's passive. Yeah. My apologies. My apologies. So it's diffusion, again, going from high to low. So it's following the gradient, so it doesn't need energy. It's a good question. It's a protein, a certain type of protein that is attracted to that molecule. There's literally hundreds and thousands of them, but the point is just that it can't do it on its own. It's not able to get through the membrane without something else helping it. It's a good question. Okay, one more piece and then we'll stand up for a break. After facilitated diffusion, there's one more good method way of transporting, and that is called vesicular. A vesicle. What is a vesicle? It's a bubble-like membrane that grabs and moves larger particles through the membrane. It's like the little spaceship that allows it to transfer between one and the other. Right? Again, it can't do it on its own. It won't fit through the channels in the membranes that are already there, so therefore it needs a special vehicle to carry it in and out. And that's what these vesicles are. And there's three that I want you to know of ways we move things. First, we have endocytosis. Endo means inside, right? So this is a way of bringing processed materials into the cell. And there's two of them. It's phagocytosis, which is cell eating. We're actually getting solid particles and bringing them in. Then we have penocytosis, which is cell drinking, which would be bringing water or other liquid droplets into the cell. You with me? Again, endocytosis, bringing in. Opposite of that would be exocytosis. Exo means what? Outside. Yeah, the outside. An exoskeleton on a bug is the outside skeleton, right? So exocytosis is we're moving materials out of the cell. Pretty straightforward. I forgot to mention receptor-mediated endocytosis. Again, this would be where usually they would be solid pieces. So this would be phagocytosis, where I'm bringing a solid something inside, but it has to first be grabbed by a receptor. It has to attach to that receptor. Then the vesicle comes around it and brings it in. So again, it can't get in on its own. It's like somebody who's a member at a country club. I can't go play that golf course on my own. I have to go in with that member. Does that make sense? A lot of exclusivity around here. Excellent, good. Stand up and stretch. We're doing good. We've got 15 minutes still. We're doing good. You're stretched. Make sure you're with me, because we're almost done. Because the last piece, you guys have heard all of these things about the pieces of the cell. You can sit down when you're ready, but just so you know, it's there. Yes, ma'am. Endocytosis, exocytosis. Well, endocytosis has three pieces: phagocytosis, penocytosis, and receptor-mediated endocytosis. And then exo is on the end. Because those three are bringing in, exo is bringing out. Okay, let's talk about the cell interior. All that conversation was simply to talk about the cell membrane. Is that not crazy? Okay? All of that was just talking about the outside, because that's a very important piece, right? How it interacts, how things go in and out, very important. But now we're going to focus on the inside of the cell. We'll talk about the actual organelles that are inside the cell, the things that actually do the work of the cell. Before we get to organelles, though, we have to mention the cytoskeleton. The cytoskeleton is a network of protein filaments and cylinders that help make up the inside of the cell. It organizes it. It gives it shape. It supports things. It helps things move inside the cell. And there's three different pieces of that cytoskeleton that are there. They are microfilaments, intermediate filaments, and microtubules. They're all kind of the same thing. They're just different sizes. Are you with me? So a microfilament is smaller than an intermediate filament that is smaller than a microtubule. You say, wait, but Mr. Hess, these are both micros. Both micros. Are you comfortable a tubule is bigger than a filament? A tube is bigger than a little filament? That's how I remember it. Okay? So again, all doing similar pieces. I'm not really worried that you understand all of the intricacies of what each of these pieces do. They're giving structure. They're giving shape to the inside of the cell, allowing things to be attached to them. So they're like anchor points, as well as just allowing things to move around in the cell itself. Because the cell is not just a blob of goo, right? Like it actually has structure and function to it. Let's continue on with these organelles then. These are the internal structures in the cell that actually have a function. They carry out a specified metabolic task. A question? Yes. We're going to talk specifically how some of them work. The ribosomes and the mitochondria in particular, we'll talk in big detail about how they work. Some of the others, we're not as interested in at this level. You go on? Sure. Yeah, you'll learn much more about them. Yeah. Okay, so let's dive into these organelles. The first one is the nucleus. And we give it primacy. We talk about it first because why? It's the biggest. I would agree. Most important. Why is it the most important? It's got the DNA, right? And the DNA is the cookbook for everything your body needs to survive, right? Every protein that makes you up, and there is a humongous list of them, the instructions to make that protein are in the DNA, right? DNA, the nucleus obviously holds all of that. That contains the chromosomes, which has the DNA. It controls replication of that material. So there's actually a process where that material is reproduced, copied, right? And passed on to your ancestors. And that nucleus is enclosed in what we call a nuclear envelope. It's basically like its own little mini cell, right? So here is the nucleus, and this is the nuclear envelope along the outside. And that nuclear envelope has little pores, little openings that are in them. We call them nuclear pores. Again, we're not creative anatomists. We just name it what it is. And that allows some of that genetic material to come in and out with that transfer RNA and messenger RNA. We'll get to that. Okay? Question. I had a question. Did I answer it? Somebody had their hand up. No, you're good always. All right. Okay, let's keep going. Past the nucleus, we're going to talk about the endoplasmic reticulum, the next. And that's basically a system of channels, and they call them cisternae. Again, it's this Latin word for channel, that is enclosed by a membrane. We've got two different types of these endoplasmic reticulums, the rough and the smooth, like literally based upon how they look under a microscope. Okay? The rough ones, they're rough because attached to these endoplasmic reticulum are little things called ribosomes. And the ribosomes we'll talk about in a minute, they create proteins, right? Their whole job is to synthesize proteins. Okay? So the rough ER are covered with ribosomes for protein synthesis. The smooth ER, which again, these are the ones without the ribosomes, one of their big key pieces, they do other things as well, but they help regulate the calcium level in the cell. They also help move things around. They package. There's, again, lots of things these guys do. They help make steroids, other types of fats, right? But again, the key piece I want you to focus on is that they help regulate the calcium concentration in the cell. So rough and smooth, and they show you here's smooth, right? And here's rough. They have the little dots all over them. What are the dots? Ribosomes. Okay? So let's talk again about ribosomes. Again, their key thing that they're doing is creating proteins. Please don't just memorize protein synthesis, because I may not use those words. What does it mean to synthesize proteins? To make them, to create, right? So don't just memorize protein synthesis. It makes proteins. It's basically reading the code from the DNA and producing the actual protein. We'll talk about this in much more detail in the future. Assembling those amino acids. Remember, the amino acids are the building blocks, the monomers, to make the polymer, the protein. Some of them, most of them are bound to the endoplasmic reticulum, but there are some that are just kind of floating around inside the cell. They're not attached there. We've got the Golgi apparatus. This is another series of these cisterns, again, these channels. And what I want you to be comfortable with, what those do are two things. They package cells, right? They're packaging them. It's like the UPS store in the cell, right? It's going to package these different products into vesicles, right? Vesicles are those little bubble, right? Little vehicles to move things in and out of the cells. So we call the packages that come from the Golgi apparatus the Golgi vesicles. Makes sense, right? And number two, it's involved in carbohydrate and glycoprotein synthesis. So it helps taking and breaking down carbohydrates, making more complex carbohydrates from some of the glucose or some of the pieces that are present there, as well as glycoproteins. These are the combination of sugars and proteins together. Let's keep going. Again, lots of pictures that are present there. Lysosomes. Does anybody know what it means to lyse something? Sorry? To cut. to break. If I spray Lysol, what does it do? It kills bacteria. It cuts it, breaks them down, right? So lysosomes do the same thing. In particular, they're enzymes that they focus on digesting or breaking down things inside the cell, like getting rid of some of the waste products or extra pieces that we don't need, extra organelles or extra parts of the cells. They're also part of something called autolysis, where if I'm producing too many of these cells, the cells can actually like kill themselves, cell suicide, which doesn't mean they're depressed or something. It just means they're unneeded, so the body will get rid of them. And these lysosomes will be what will help with that.
So the body tells them like they're not needed.
Right. It's keeping this homeostasis. Maybe for some reason the factory that was producing these cells got too, it's like, hey, calm down. All right, let's just get rid of those and we'll keep going.
But it actually kills itself.
Sure, yeah.
No desire to
I looked at the wrong time. All right, good. We did a good job. We only had a couple more pieces. Make sure you check out the last couple. We finished with lysosomes. We got peroxisome. Inclusions? That's just things that are stuck inside the cell. Nope, no, that's the actual name of it. All right, friends, adios. Yes.